The Laboratory of Cancer Biology and Cannabinoid Research, Department of Biology, Technion Israel Institute of Technology, Haifa, Israel. The effect of cannabis plant extracts on head and neck squamous cell carcinoma and the quest for cannabis-based personalized therapy. In this research, we tested the effect of various cannabis extracts on HNSCC cells, characterized the chemical profile of those extracts with antitumoral activity, determined their antitumoral mode of action, tested their impact on non-cancerous cells, and examined endocannabinoid receptors involvement. Phytocannabinoid extraction and sample preparation. Air-dried medical cannabis cultivars were obtained from several Israeli medical cannabis distributors. Cannabis extracts were prepared by the Laboratory of Cancer Biology and Cannabinoid Research, Department of Biology, Technion Israel Institute of Technology. 24 cannabis extracts, natural and after decarbosylation, were used for composition analysis. Extracts were classified into 3 groups according to their THC and CBD content. See Figure 1. Phytocannabinoid identification and quantification. Identification and absolute quantification of phytocannabinoids were performed using analytical phytocannabinoid standards. Fractionation of the whole extract. Fractionation of CAN296 into 4 fractions was performed and then lyophilized to dryness and analyzed. Cell cultures. 4 well-characterized human-adherent epithelial cancer cell lines were purchased from the American Type Culture Collection. Cell viability assays. Cells were seeded in 96 well plates at 7,000 cells per well. Different cannabis extracts were added in triplicates of concentration. Cell apoptosis assay. Cells were seeded and treated with cannabis extracts as described for cell viability assays. Apoptosis was assessed. Cell lysis and Western blot analyses. Cell signaling technology. RNA extraction. RNA was prepped. Real-time quantitative emulation. Linear comparison of gene receptor expression. Statistical analysis. Statistical analyses were conducted. Synergy finder. Drug combination dose response matrix data were analyzed with the Synergy Finder web application multiple comparisons test. Results are the heterogeneous composition of cannabis extracts. We quantified the phytocannabinoids profile of each extract. The 17 most abundant phytocannabinoids quantified in the 24 extracts are presented. See Figure 1. Decarboxylated high CBD extracts are the most effective in inducing HNSCC cell death. To examine the effect of the 24 extracts on the survival of HNSCC, we then used 4 well-established oral origins of SCC cell lines: SCC4, SCC9, SCC25, and Cal27 based on the initial screening. All extracts in the decarboxylated CBD type group and 1 extract in the decarboxylated THC2 CBD type group were significantly effective against HNSCC cancer cells. Figure 2A and 2B. Cannabis extract CAN296 shows apoptotic effects in HNSCC. To examine the mechanism of cancer cell death induced by the cannabis extract, we treated SCC25 and Cal27 for 12H with CAN296 extract at different concentrations. See Figure 3A and 3B. Based on the initial screening, all extracts in the decarboxylated CBD type group and 1 extract in the decarboxylated THC CBD type group were significantly effective against HNSCC cancer cells relative to the other extract. Treatment resulted in increased cell death through apoptosis, both early and late, in a dose-dependent manner. See Figure 3C and 3rd. Cannabis extract CAN296 is more selective towards HNSCC cells compared to non-cancer fibroblastic cells. We examined the selectivity of extract CAN296 to HNSCC cell lines by comparing the normal cells. SCC25 were significantly more sensitive as treatment resulted in significantly higher percentages of cell death, approximately 90%. See Figure 4. Endocannabinoid receptors expression in HNSCC cells. To better understand the mode of action of cannabis extracts on HNSCC cells, we analyzed the mRNA levels of 7 common endocannabinoid receptors in the 4 cell lines using real-time qPCR. 3 of the cell lines expressed high levels of CNR1, and all except SCC4 demonstrated very low expression of GPR55. Receptors CNR2, TRPA1, and TRPM8 were expressed in a mixed pattern across the cell lines. TRPV1 was the primary receptor expressed in all the cell lines, and TRPV2 was highly expressed in SCC4 and moderately expressed in all other cell lines tested. To identify endocannabinoid receptors' involvement in the cytotoxic activity of the cannabis extract, we pre-treated SCC25 cells with different inhibitors. Identifying the active molecules in CAN296 responsible for the apoptotic effect. To identify which active molecules in the CAN296 extract were responsible for the apoptotic effect on the HNSCC cancer cells. Preparative ultra-high-performance liquid chromatography was used to fractionate CAN296 extract into 4 distinctive fractions. Each of the 4 fractions had a unique dominant cannabinoid content. Fraction 1 was CBDV dominant, Fraction 2 was CBD dominant, Fraction 3 was THC dominant, and Fraction 4 was CBC dominant. The fractions were normalized to match their concentration in the whole extracts by diluting each fraction to match its dominant cannabinoid concentration to its concentration in the whole extract, and SCC25 cells were treated with each fraction or different combinations. Figure 7. Combination of CBD with CBC or THC results in a synergistic effect on HNSCC cell viability. To better understand the relationship between CBD, CBC, and THC treatment, that is, whether there is an additive or synergistic correlation, we tested combinations of synthetic cannabinoids CBD, THC, and CBC. We designed a study of a dose-response matrix for CBD, CBC, and CBD THC and examined their effect on the viability of SCC25 cells. See Figure 8. We further examined the effect of increasing concentrations of 2.8 grams ml of CBD and CBC combination at the synergistic ratios. Discussion. In this research, we compared the cytotoxic effect of 24 representative cannabis extracts on HNSCC cells. We found that decarboxylated CBD-type cannabis extracts led to a significant increase in cell death, and this effect was consistent throughout all HNSCC cell lines tested. The decarboxylated CBD-type CAN296 extract was chosen for further characterization. We found the cytotoxic effect to be dose-dependent, acting via the induction of apoptosis. Moreover, the cytotoxic effect was specific to HNSCC cancer cells, as similar concentrations did not induce apoptosis in fibroblasts, falling in line with previous reports indicating the selectivity of the apoptotic effect. By nature, cannabis extracts display a wide variance in their metabolomic composition, with conflicting reports of the anti-tumoral effect. Consequently, active plant ingredients must be identified and standardized for adequate clinical anti-cancer treatment. For that purpose, the chosen crude extract CAN296 was fractionated, and different combinations were formed, normalized, and tested for cytotoxicity on SCC25 cells. While the CBD-dominant fraction was able to induce a cytotoxic effect on the cancer cells by itself, potency was notably less than the whole extract. emphasizing that the cytotoxic effect of decarboxylated CBD-type whole extract on HNSCC cells is higher than pure CBD. Moreover, the group of extracts with the highest CBD content possess the highest cytotoxic effect. Extracts with moderate CBD content had a moderate cytotoxic effect, as seen with extract CAN 298. And extracts with minimal CBD had a neglectable cytotoxic effect. When the CBD dominant fraction was combined with any of the CBC or THC fractions, the cytotoxic effect matched that of the whole extract, pointing out an entourage effect between CBD-CBC and CBD-THC, a known feature of cannabis extracts. Interestingly, in the decarboxylated CBD type group, both CBC and THC were present in low amounts of 2.5, 3.3%, with a ratio to CBD of 1 to 15, 1 to 20, respectively. Higher amounts of both molecules were needed to achieve the full cytotoxic potential equivalent to that of the extract. Synergy model testing confirmed the combination between CBD-CBC or CBD-THC is synergistic, with an optimal ratio of 2 to 1, respectively. While previous reports of THC-CBD synergy demonstrated reduced tumor growth, some studies suggested combining THC with CBD mainly modulated the adverse effects of THC. This research points out CBD as the main API, and combining it with either CBC or THC at a ratio of 2 to 1 maximizes the cytotoxicity on HNSCC cells. Considering the adverse psychotomimetic effects of THC, there is a clear advantage for favoring the CBD-CBC combination over CBD-THC for novel treatments for HNSCC. To the best of our knowledge, only one previous report pointed to the interesting biological activity of CBC and its potential for medical use. With the mechanism yet to be discovered, a synergy between CBC was found to enhance the cytotoxic effect of THC in treating urothelial cell carcinoma. Our research found CBC to enhance the cytotoxic effect of CBD, establishing additional support for the phenomenon of the entourage effect in phytocannabinoids. CBD has been previously reported to affect cancer cell growth through different pathways, including the induction of cell cycle arrest at the G0-G1 phase, increasing the levels of pro-apoptotic signaling proteins, increasing the levels of cleaved caspase-3 and -9, or by activating the mitochondrial apoptotic pathway. The combination of CBD with CBC was more effective than CBD by itself. CBD can bind multiple targets, and CBC was shown to bind TRP channels. Our finding aligns with publications that demonstrated that some cannabis extracts enriched with specific cannabinoids were more potent agonists of TRPA1, TRPV1, TRPV2, and TRPM8 receptors compared to these same pure phytocannabinoids individually. Yet, it is not clear whether the cytotoxic effect of CBD and CBC combination is the result of one cannabinoid enhancing the activity of the other, or from hitting different signaling pathways at once. We examined RNA expression in order to characterize our relevant cell lines in terms of gene expression of cannabinoid receptors, as well as receptors known to mediate cannabinoid signaling. A high expression of CNR1, TRPV1, and TRPV2 was noted. In contrast, the expression of CNR2 was very low, which falls in line with previous works suggesting that CB2 is expressed mainly by immune cells. GPR55, TRPA1, and TRPM8 receptors expression was not consistent in all cell lines and ranged from medium to low. While CBD is an antagonist or a negative allosteric modulator of CB1, CBD and THC can activate TRPV2, TRPA1, and TRPM8 receptors, and both CBD and CBC have been recorded to activate TRPA1. Nevertheless, in this study, All attempts to block the cytotoxic activity of CAN296 extract by inhibiting CB1, CB2, GQ, GPR55, TRPV1, and TRPV2 failed to reduce the cytotoxicity of CAN296 treatment, suggesting that the mode of action of the extract on HNSCC does not involve these specific receptors. Conclusions. This research suggests using whole cannabis extracts, which are decarboxylated CBD-rich, to induce cancer cell death. In recent years, many cancer patients have been treated with cannabis extracts as a palliative treatment. Based on this research, these chemovars can provide additional anti-cancer properties in addition to the palliative effects that cancer patients can benefit from. Furthermore, we recommend enriching extracts with CBC to reach a CBD to CBC to THC ratio of 2 to maximize the cytotoxic effect on HNSCC.